Okay, so yeah, apologies for the delay and the technical issues, but hopefully this next kind of sort of 30 minutes will give you an insight into the challenges and the career opportunities for people that want to explore making a medicine. So if we think about the process itself, in essence, from discovery, from that initial moment in the laboratory to the patient, normally takes about 12 years, and it normally costs in the region of £1 billion. And so there's 4 stages to the process that I want to share with you today. And therefore, that'll highlight some of the different career options that are within those different stages. Whether it's right from this word go in terms of the laboratory, or whether it's at the more clinical facing end of actually delivering that medicine to the individual that needs that medicine. So I think the first thing to highlight is that indeed making a medicine, successes are rare. So I'm a neuroscientist and involved in Alzheimer's disease research. And it's a little bit embarrassing to have a look at these numbers and say that out of 123 attempts, only 4 medicines have made it to approval. And those 4 actually don't actually impact on the disease itself. They are more treating the symptoms of the disease. And some of the statistics are the same for melanoma and also for lung cancer as well. So again, having that eureka moment in the laboratory doesn't necessarily at the other end of going to share today. So if you think about discovery, this is the first stage, thinking about maybe highlighting or trying to pick a gene or a protein instrumental in a disease process. So to find out something that goes wrong in the human, potentially or a drug with this. Will be blocking an essential receptor. Disease. But a new.
Hi, sorry to interrupt. You're very laggy.
Those interested in the, but if you look at these cells, colored black and one's colored blue, as a side farm, we're interested in looking at the differences in these cell types. What makes one the color blue? Predisposed to actually disease situations. Okay, in terms of hello, Sasha?
Your audio is very laggy, so would you mind turning off your video? Because that might be affecting the internet connection. Hello?
Hello?
Hello, can you hear me?
So I hope you can still. Okay, I'm just. Yep, okay. And come out. I don't know if you can.
You can keep the screen sharing on. It's just your video, because that might be causing the lag. Okay.
Yes, but I don't have that option on the screen, so I need to stop sharing just to be able to bring that option up. Okay, turning camera off.
We can't see your screen at the moment.
How about that, Sasha? Is that any better?
Not yet.
Is it coming through?
Yes, now we can see it. Perfect. Thank you. Yeah, audio is still fine.
And how about audio? Okay, perfect. Okay, so yes, where we were, we're thinking about trying to find a target, in essence, for us to make our medicine, whether that be a gene or whether that be a protein. And for the purposes of the kind of sort of going forward in this session, we're just thinking about a protein. What proteins are of interest from a disease perspective in order for us to make a molecule that is going to actually target that. And we touched on before the kind of technical issues, we touched on thinking about nerve cells and what the differences would be maybe in a disease cell and trying to uncover what that means for that cell and how we could potentially make a molecule to change that function. So again, just to recap, hopefully most of you are interested in biology and you have an interest in human biology. So just the basics of a gene and a protein are listed here. In essence, it's the unit of hereditary for genes. And then the protein are these fundamental structures that in essence are in every cell of the body. And they have a whole host of functions to play. So again, for going forward, we're going to be thinking about proteins. So this is just a schematic on the left-hand side. It's a picture, and it's don't need to worry about the NAV 1.1, which is the kind of protein name, but it's just here to highlight what did we want to know about the proteins we discover. Well, one, we want to know the structure of these proteins. So again, you can see in that cartoon, nicely illustrated, there's different subunits that come together, this alpha and the beta subunits. And then indeed they are sitting within the plasma membrane. So you can see the cartoon of the plasma membrane. So we know where in a cell this sits, but we also know a little bit about the structure. And potentially then that gives us an idea to function. And so this one that's highlighted here is a voltage-gated sodium channel. And that's involved in allowing sodium to enter into a cell and allow, in my case, a neuron to fire an action potential. So that tells us a little bit about structure. Then going across to the kind of cartoons of the different organs of the body, we want to know where in the body these proteins are expressed. That'll give us an insight into potentially, one, the therapeutic benefit. So it might be a particular cardiovascular disease that you're interested in, or a particular neurological disease that you're interested in. And again, that'll give you insight into where best to target this particular protein, but also it might give you insight to side effects. So again, if this protein is expressed throughout the body, it's thinking of ways in which you could maybe minimize side effects if you're particularly only interested in solving a cardiovascular problem. So we've discovered something of interest, a protein that's involved in a disease process. Now we need to start thinking about making that medicine. So there's two ways we can go about this. You can target an existing molecule, and here there's a stack of books which signifies a library of compounds. Most research laboratories and pharmaceutical companies have libraries of compounds that started their journey potentially to be a blockbusting medicine, but for whatever reason didn't process or didn't proceed to fully approved medicine. So you can go back and look at what we have on the shelf, and compound screens run to the tens of thousands of compounds looking to see if we can have any activity against that protein that we found in our discovery phase. On the other hand, we might be actually thinking about making a new medicine. So it's thinking about chemistry, thinking about making or synthesizing a new molecule that we think can have activity against our receptor. And again, from phase 1, that structure of the protein we're interested in will give us an insight into, in essence, fitting our molecule, fitting our drug of interest into the receptor. And we can think of it as a lock and key. We've got our target protein as the lock, and potentially we've got some new molecules here. And just using the color schematic, it's the blue one that's going to fit. All the keys look very similar, but in essence, it might be that only the blue one is the one that has the activity against our target protein at the correct concentration that we're interested in. So again, it's thinking about medicinal chemistry. It's thinking about how we can change that molecule to make a better fit, but also to improve the actual impact on the activity of the target protein, whether we're wanting to switch that on or whether we're wanting to switch that off. So we've got our molecule of interest against our target protein. Now we need to make it into a medicine. So just here are some of the types of medicines you might be familiar with: tablets, syrups, gels, and an inhaler. So there's many ways in which individuals can take medicines, whether that's through the oral route, or whether it's through injection, whether it's through rubbing on gels or creams, or whether it's through inhalation. Each of these medicines is made a very different way. So you have to start to think about potentially the population of individuals you're trying to treat, whether that be the young or whether that be the elderly. The tendency here is to steer clear of tablets and capsules. From the younger perspective, difficulty swallowing, and you don't want choking. And that's the same for the elderly. Although again, the elderly may struggle to actually pop the capsules or pop the tablets out of their packaging. So again, it's starting to think about the formulation of the medicine you're making with a view to then your target audience. And again, there's different ways in which you would then start to make that medicine. Different constituents go to making up a tablet as opposed to a gel or indeed a cough syrup. So we've got something of interest, a molecule that targets our receptor. We know in essence that it fits and has activity. We now need to start to ramp up the testing. And this starts very much at a preclinical phase. And that's thinking about potentially using animal models, or indeed now more and more, we're using human models. So we're using stem cells in the laboratory, starting to look at whether it be the brain or whether it be the heart that we're interested in. We can start to generate 3D models and start to look at the effect of the drug on these particular human models. And this all starts to build up a better picture of how the drug will work in vivo, starting to understand how all the different systems of our models come together to actually give an overall benefit, if that's what we're looking for, and also might give us indication of any side effects or detrimental effects that we hadn't really seen in the test tube. So these are mainly laboratory based. So following preclinical, we're then into clinical trials. And these are staggered through different phases, and some of them you might be aware of. So phase 1, which if you've been watching the news, COVID-19 has been recently going through, is just getting a small number of healthy volunteers, giving them the drug or medicine that you've made, and then analyzing what it does to their body. But also it's thinking about what their body does to the drug. How well is it absorbed or metabolized? And indeed, does it get in sufficient concentrations to, in essence, the target organ? So we know the brain is very privileged with the blood-brain barrier, but does enough of our drug get into the brain for us to be confident in terms of going forward and trying to treat patients with a specific disease? Then we're ramping up to phase 2, so starting to increase the number of individuals that we're testing. We're keen to find out what the population effect is rather than these just smaller cohorts that we started with in phase 1. And again, it's blinding these trials. So we're starting to take away the knowledge of what particular medicines are involved for those individual researchers. They have no knowledge of whether they're giving, in essence, the placebo, or indeed whether they're actually giving the real drug. And then that allows to gives you a little bit of robustness and confidence in the data that you've generated. A lot can be said from an individual knowing that they're giving them the real drug, and the individual taking it can pick up on those sort of unconscious signals. So that gives us a little bit more information about the safety and, in essence, the effectiveness of our new medicine. Phase 3 builds it up again now. We're now starting to look at maybe multi-center investigations, maybe looking to see in America, in Europe, in Asia, what are the different effects of this medicine and how they're possibly leading to benefit. There may be some side effects, but again, it's starting to think about the whole population or the global population and teasing out how effective this drug really is for the patients that are, in essence, going to be taking it. So now we're looking at, in essence, the disease patient cohort. End of phase 3 tends to be when people start going knocking for the FDA and the EMA to try and get approval for marketing their drug. But it doesn't stop there. So we need to continue on and keep up looking and watching what these drugs are doing. And that's called, in essence, phase 4. And it's monitoring the drugs to get some longitudinal data. So looking at the long-term effect of taking these drugs. That's not something you can gain from the clinical trials because they're very rapid and quite quick in terms of the procedures to try and get the effective data to then get the FDA to approve it. So we need to monitor the long-term effectiveness and the impact of, in essence, patients' quality of life. So we've discovered, we've made, we've tested, and now we're seeking approval. So again, the two main bodies here are the Food and Drugs Administration, the FDA, and the European Medicines Agency. What they're looking at is, in essence, looking at what you're trying to treat. So taking the target condition, whether that be a cardiovascular disease or a brain disease, and seeing what's currently available. Then they're assessing the benefits and risks of the clinical data. So again, it's a balancing act. So we mentioned that there may be some adverse side effects or some adverse events. How does that overweigh or outweigh the benefit? Some of you may be aware of chemotherapy has very bad side effects, but in essence, they're actually, outweighed by the benefit to the patient in terms of eradicating the cancer. And again, it's thinking about strategies for managing risk. How are we going to monitor this drug going forward? And indeed, what individuals are going to be involved? Is that going to be the doctors? Is that going to be the pharmacist? Is that going to be the nurse? Who's going to be kind of responsible for flagging up any sort of ongoing issues with people taking this medicine? So it's a whole kind of spectrum of individuals and the level of responsibility that they take in terms of managing risk. So we've got that letter from the FDA or the EMA to say that we're happy to approve your medicines. Then it goes onto the shelf, and it typically goes onto the shelf in a pharmacy. So I'm sure we've all been into a pharmacy and collected your medicine, whether that be over the counter or whether that be something that's on the shelf. And again, thinking about those medicines you may have taken, thinking about the packaging. So again, it's about getting the packaging right, getting that information on that box that potentially the individual is going to receive to make it clear to them what the medication is, what it is they need to do with it, and in essence, what potentially might happen going forward. And some of that information is contained on the box, but also some of it's contained on, in essence, the information leaflet, and that aids adherence to medication. So some of you may have taken paracetamol, may have taken Nurofen. Have you actually emptied the box to see everything that's inside? You normally find a piece of literature like this, and it gives a whole host of information. Tells you what they're used for, what you need to know before you take them, how you should take them, possible side effects to look out for, and also important information about storage of medicines. And again, it's not something we're all readily aware of in terms of where we should store medicines depending on the actual medicines itself. So the storage instructions, which are key. So we've got 1, 2, 3, and 4. So that's the kind of 4 stages which I wanted to touch with you today. But in essence, it's just to give you a little bit of insight into what I've come up with is 62 different career opportunities within the drug discovery program. Whether that be right back at stage 1, a pharmacologist, a laboratory scientist, maybe a statistician, going right through making synthetic chemists, lab scientists, toxicologists, right through to, in essence, the frontline healthcare professions that you'll probably be hearing a little bit more about in this conference later on today. So I hope that's given you a little bit of insight. What I want to do, and hopefully this will work, is share with you a video of us doing it the Reading way. So at the University of Reading, we're very privileged that we've actually taken something from the laboratory into clinical practice. And I don't know if any of you are aware of the recent news items about cannabis-based medicines, but this is in essence Epidiolex. It is in essence a medicine made from cannabidiol or CBD, the non-psychoactive ingredient in cannabis, and it's used to treat epilepsy in younger children. So hopefully this video will play. It doesn't seem to be playing my end, so I'll tell you the story myself without the use of the video. So in essence, we started that kind of journey that we talked about in terms of discovery with different constituents of cannabis and looking at the non-psychoactive ingredients. And we found very early on that they were effective at reducing epileptic seizures in some of our animal models. And we then partnered up with GW Pharmaceuticals, a pharmaceutical company, and they supported our basic lab research to build that evidence-based data to then support a full application to the FDA. And now it's approved in America, but it's also approved in Europe as well. So while I said it was successes are rare at the very start, we've been part of something at the University of Reading that's actually succeeded and gone through all those hurdles and challenges. So that's it from me. Hopefully in this brief session, it's given you a little bit of insight into what's going on in drug discovery and also thinking about potential career opportunities in that pipeline. Everyone contributes their little part. Their little bit of expertise is an important cog in making that medicine, which is fundamentally going to then deliver the medicine to those individuals that need it. So thanks for joining us for the session. Apologies for the technological hitches.
Yeah, there are already some
if anyone's got any kind of questions they
in the
want to
chat, I
ask,
can read them out.
feel free
There's a question that's,
to use the chat
would you say
function.
going into pharmacology requires more interest in chemistry as a subject than medicine?
Yeah. I wouldn't say, in essence, chemistry would be most important for pharmacology. I think it's an interest in medicine. It's something that's going to drive you towards that success. I think biology and chemistry would be kind of ideal. A lot of the pharmacology is more based on biological systems, so thinking about the physiology of the system.
Next question
and
is, how long did it take to get
then lead to
Epidiolex
generating a new medicine? So Epidiolex was, in essence, 10 years in the making. So it took us 10 years for that initial kind of eureka moment while we were beavering away in the laboratory to now it being approved in America and Europe. So 10 years. So I think you can be involved in many ways. There's been the recent call in terms of trying to get patients or healthy volunteers to take part in drug trials. So if that's something that interests you, then they're always looking for people to try drugs out on. But in essence, there's also things that you can get involved in terms of being patient voices. So again, if you're taking any medications or have taken paracetamol, ibuprofen, or maybe you're suffering from asthma, you can join those patient sort of groups and be an advocate for, in essence, improving medicines. We as scientists always need to listen to the
Next
people that
question
are
is kind of
taking the
similar. With
medication
regards to
to find
pharmacology,
out
what
how
sort
we
of
can
work
make it
experience
better for
options
them.
are out there?
In terms of pharmacology, I think if you can get any experience in a laboratory base, that would give you a little bit of insight into taking it forward. You might be aware that a lot of this stuff now is coming out of the laboratory and being much more big data. So the options there are, in essence, looking at using computer science to model biological systems. And a lot of pharmaceutical industries are now crying out for, in essence, graduates to have a little bit of math skill or a little bit of coding, computing to actually put towards pharmacological problems. So in essence, work experience can range from lab science, so getting into a university lab. I know there's very summer sessions or summer vacation studentships that are available to go and join universities. But in
There's a
essence,
question
just
on step
shadowing someone and
2
getting a feel
making.
for
Does the lock
what
and
it would
key
be like
model
day
for
to
the new
day.
molecule and protein work like an enzyme substrate complex? Could the target molecule denature?
So yeah, interesting question, and thanks for sending that in. So in essence, yeah, it is a bit like the enzyme and substrate complex, but in essence, you're not in essence actually then denaturing or changing in essence the metabolism of the substrate. So in essence, if we're taking the substrate to be the target molecule, in essence, all you're doing is changing the function of that, or it might simply just be sitting in and plugging a hole. So in essence, the one I showed was a sodium channel. A lot of the drugs that impact on sodium channel function actually just sit in the pore and block that sodium going through. So while enzyme substrate complexes are more or show higher binding or higher integrity, the
Next
drug
question is, do you know
molecule
much about the
interactions isn't
Tamiflu
as tight as an enzyme
controversy? How
substrate
can we avoid
complex.
things like this in the future?
So I'm not sure about the Tamiflu controversy that the question is referring to. I guess if they can maybe add another or add a subsequent question just to indicate what the controversy they're indicating. If it's around potential adverse events that have not been picked up until we've got to drug approval, I think, and this might not be answering the question, but I'll answer that one anyway, is that in essence, what we're now using is changing our tone from animal models to human models, starting to very early on look at what the human does in terms of adding medicine, adding drug, and thinking about what's going on there. And that is starting to have much more success from us translating what we see in the laboratory to what we see in the clinic. And I think that's key going forward and thinking about modeling human data rather than relying on animal models, which give us a little bit of insight, but again, that unfortunately gives us a lot of drop-offs in terms of something working very well in vivo in
Next
an
question is,
animal
as a
that
future
doesn't
doctor,
then correlate with
what routes could
someone getting
one
better
pursue
when
to
they take the
further
medicine.
interest, one pursue if they have further interest in pharmacology?
Yeah, so great to see that you're still you're interested in continuing pharmacology. So obviously there'll be some basic pharmacology within your course curriculum. and I'm not a medic, so I wouldn't know the full details of that. But if you wanted to continue on, you can become a clinical pharmacologist. So you can actually do your training and then do further training on the job, if you like, to then become really focused on the pharmacology and be a clinical pharmacologist and an expert in the field of pharmacology. And that's more at the clinical phase. So thinking about all those different medicines, it might also be involved in taking some of those clinical trials forward. So one of the roles there that a medic could play is actually maybe monitoring clinical trials or being involved as the lead researcher. So there's a whole host of opportunities to continue your interest in pharmacology. But day to day as a doctor, pharmacology will probably come up time and time again, and you'll think
The
back
next
to your
question is quite
training and
similar.
think back
So,
to
how are doctors
those
involved in the
lectures that you
process of
sat and listened
making
to
medicine?
and didn't think had much relevance. So there's very, very different levels in which they can be involved. Thinking back to the non-professional kind of question, there is the opportunity to get involved very early to say, I'm seeing my patients on a daily basis, they're taking medicine X, but it's not really working for them. And then that can send the scientists and the non-healthcare professionals back into maybe the laboratory to try and figure out what's going wrong. It might be that we have to look at the genetics of these people to try and figure out why some are responding better than others. So there could be initial flagging up to say, right, this medicine isn't working as it should. Then again, in the clinical trial process, they can be involved in actually monitoring or recruiting patients that they think are valid and meet the criteria for clinical trials. And then again, through the approval process and through that monitoring process, doctors are going to be pivotal. And in essence, they're the eyes of the pharmacologists on the ground. They'll be able to tell us, right, this is having greater effects than we saw in the laboratory, or indeed, this isn't working so well. And then it's then maybe suggesting that actually it's a combination of two
I
drugs
think
that we should be looking at,
that's
maybe
all
than the
for now. If
drug
anyone
or
has
new
any
medicine that was
questions,
generated.
now's the time because we're about to finish. We can wait maybe a bit. I think we can. There's another one. For phase 4, is that the yellow card scheme medical research, or do the pharmaceutical companies actually pay for long-term monitoring?
So phase 4, the yellow card scheme, that the pharmacists play a fundamental role in terms of logging any kind of adverse effects. In terms of who pays for that, it tends to be, in essence, pharmaceutical companies that will then access that data to find out what their particular medicines or what the adverse effects are. If they are, in essence, becoming more commonplace, then they'll then start to then think about then potentially relicensing that medication, or indeed the authorities might come in and say, actually, we're seeing this coming up time and time again on the yellow card scheme, you need to do something about it. They then might be involved in then contracting out to university labs or contract researchers to think about how they can overcome that individual problem that's been identified. Will it be a potential side effect, or will it be some kind of long-term effect on liver function, for example? So in essence, it would be the kind of pharmaceutical companies that would be picking up the tab, but they might be then
going back to the Tamiflu
working and collaborating
situation,
with basic
I looked it up and it
researchers at
says that
universities or laboratories.
the drug may pose especially serious health risks to children, causing confusion and hallucinations. So I guess the question was referring to that and how we can avoid things like this in the future.
Yes, I think that's fundamentally they're thinking about who you're making the medicine for. Children, adults, and the elderly actually respond very different to medications. That may seem surprising to us, or it may not seem surprising to us, but in essence, it's something we need to think about. So you may alter the dose, and also giving medicines to children is a very, very challenging situation to be in, because the longitudinal studies and the longitudinal data you don't really have a handle on. You don't know what that individual is going to have to face in terms of their future, in terms of maybe further down the line, in terms of they're growing, their brain's developing, a lot of their organs are still developing into the full adult form, if you like. What you're giving them as a medicine may have a very detrimental impact. And for some medications, we don't really know and have a good insight in terms of what we're doing about it. So a lot of the clinical trials and a lot of kind of the regulation has been tightened up and approved. And that's why the consequence of that is potentially maybe disappointing that we don't get enough medicines through. But in essence, if it's making better medicines and safer medicines, then as a healthcare professional, that's always going to be of benefit for you in terms of giving you that confidence when you are offering an alternative medication that it's going to be safe,
I think
and it's actually going to improve that person's quality of life.
Wonderful. Thank you so much. I guess we can finish the session. Thank you so much, Dr. Dallas, for giving us the talk, and everyone else who joined the session. Enjoy the rest of the conference.